If you're looking for insights into and best practices around high-content analysis and high-content screening and toxicity testing, you're in the right place. In this webinar entitled High-Content Screening and Analysis in Cell-Based Assays for Drug Toxicity and Genotoxicity Testing, our two speakers, seasoned HCA/HCS experts, will present two different approaches to using HCA to detect potential toxicities from cell models. I'm not John Sterling. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and in a schedule change, I'll be the moderator for this webinar. Our two speakers today, Dr. Heather Martin, Research Fellow, Leeds Institute of Molecular Medicine, University of Leeds, and Dr. Giuseppe Astori, Advanced Cellular Therapy Laboratory, Department of Hematology and Cellular Therapy at Vicenza Hospital in Italy, will discuss the use of cell-based HCS assays for early identification of compounds with adverse effects. They will also discuss how HCS cell-based assays can potentially reduce compound rejection due to toxicity in subsequent in vitro and in vivo assays. Dr. Martin will get the presentation started, but before she starts, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We'll answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. Heather, we're ready to get this webinar underway.
Hello. My name is Dr. Heather Martin from the Bioscreening Technology Group at the University of Leeds, and I'm going to talk today about the role high-content imaging can play in the detection of cytotoxicity. Drug discovery and drug development are global multi-billion-dollar industries. However, for every compound that successfully makes it to the clinic, approximately 10,000 have been discarded during the process. The reasons for failure are varied and include poor physiochemical properties, limited efficacy in clinical trials, but one of the biggest reasons for failure is toxicity. In some cases, this toxicity can be anticipated based on structure-activity relationships and computer modeling, but other times it cannot be predicted, meaning that toxic compounds are not detected early and progress into admit and clinical trials at considerable cost. The identification of hepatotoxins is a case in point. This slide shows a very simplistic outline of the pipeline of drug discovery and drug development, from an identified biological target through in vitro and in vivo testing and development, into clinical trials and then licensing for market. Within this pipeline, there are a number of specific toxicity tests, both in vitro and in vivo, that must be carried out by law before a potential new drug can be licensed. These occur downstream of candidate selection and optimization. In an effort to reduce failure rates, preliminary toxicity testing now frequently occurs earlier in the pipeline, as indicated by the arrows. There is a potential for earlier elimination of toxic compounds in combination with initial compound screening to streamline these processes further. By using in vitro techniques earlier in the process, thus failure occurs earlier and is more cost-effective. The development of high-throughput fluorescent microscopy, called high-content imaging, due to the sheer volume of data or content that can be extracted from single cells and wells, is making this a reality. These systems are automated imaging platforms, usually with a minimum of 4 channels for different fluorophores, and permit the capture of several thousand images in short time frames. Thus, large compound libraries can be effectively screened using in vitro imaging approaches that may be more biologically relevant than some of the biochemical assays available. These platforms can be utilized at all stages of the drug discovery and development pipeline in terms of identifying toxic compounds as part of lead optimization, and for those tests required by law. There have been a number of publications exploring the utility of high-content screening to identify hepatotoxins and their mechanisms of action in multiplex assays. Of particular note are those by O'Brien and Pearson, respectively. These assess a number of endpoints, including membrane integrity and mitochondrial health, and compared the ability of these platforms to detect known hepatotoxins when compared to the conventional cytotoxicity tests. Indeed, the high-content approaches used had over 90% specificity and sensitivities of between 50 and 90%, depending on the endpoint assessed. So the utility of high-content imaging for the detection of cytotoxicity has been established. However, these assays are designed solely for toxicity detection, utilizing all available imaging channels in the systems used. And whilst they should reduce the number of compounds that fail in vivo ADMET studies, as well as streamlining the testing procedure, they do not fully address very early compound failure. This type of assay is indeed relevant at the points indicated by the two rightmost arrows on the diagram. And as I've just said, there's an opportunity to use high-content imaging to reduce compound failure earlier by screening with initial screening for active compounds, as shown by the leftmost arrow. Indeed, today I'm going to talk about an assay that has been designed to be used in this role as part of initial compound screening to identify compounds that may fail later. In other words, an assay that can be used to generate toxicity data alongside activity data at the beginning of the pipeline, as highlighted in red. This work was published in PLOS One earlier this year and is very much a proof of principle study. Briefly, this assay utilizes a nuclear stain in the blue channel and a cytosolic stain in the far red channel. Such stains are routinely used as part of high-content screening to determine subcellular localization and subsequently are unlikely to impact on the primary screening output measured in the remaining channels, which will vary between facilities. This assay was developed using an osteosarcoma cell line, U2OS cells, that are very amenable to high-content screening as they form flat, evenly spread monolayers. Other cell lines could be used with this assay. For example, HepG2 cells used by O'Brien and Pearson, or other cell lines appropriate to the primary screening endpoint, e.g. a neuronal cell line if exploring Alzheimer's therapeutics. A small library of 330 compounds with a diverse range of structures rich in natural-like products was used to test the assay, which uses 6 cells that are then fluorescently stained. The images were taken and collected using the Perkin-Elm operetta, a wide-field high-content imaging system equipped with a plate hotel. This slide shows in more detail a flow-through of the experimental setup. The U2OS cells were plated 24 hours before the addition of the test library at a concentration of 20 micromolar using a liquid handling platform with 40 compounds added to each plate in duplicate. The plates were then fixed in paraformaldehyde following either 24 or 48 hours exposure to the test library and permeabilized, before being stained with DAPI, TOTO3 iodide, and an anti-phosphorhistone H3 antibody. 11 fields of view per well were then imaged at 20 times magnification and transferred to the Columbus software for analysis. This slide shows a sample image from a single field of view of a vehicle-treated well. The top left image shows all 3 channels together and then separated into the blue channel, top right, showing DAPI staining, the far red channel, bottom left, showing TOTO3 iodide staining, and the red channel, in this case with yellow false color to distinguish it from the far red channel, showing phosphorhistone H3 staining. The images generated were then analyzed in the Columbus software, looking at simple measures of cell number and morphology to distinguish cells that varied from normal cells, what I will now term abnormal cells. Firstly, the nuclei of all the cells in the image were identified with the fine nuclei algorithm based on the DAPI channel, as seen in green, before those on the periphery of the image were excluded, the red cells. Next, the extent of the cytosol of each cell was identified based on TOTO3 iodide staining that stains RNA under the conditions used here, that is to say fixed and permeabilized. In live cell assays, TOTO3 iodide is used in a different manner to assess membrane permeability. In the box shown in a zoomed-in image outlined in yellow, it can be seen that the shape of the cytosol is accurately mapped by the software. Once these regions of the cell have been identified, the size and shape in roundness terms of both the cytosol and the nucleus were measured, together with the intensity of DAPI staining, on an individual cell level. From this, the values for vehicle-treated wells were compared to those treated with doxorubicin or Taxol, which allowed the definition of normal and abnormal cells based on cutoffs. Taxol and doxorubicin were selected as potential positive controls, whilst as not being true cytotoxicants, they are well documented to have multiple cellular effects, including inhibition of cell cycle and induction of apoptosis. And by not using pure cytotoxicants, the assay's ability to detect subtle and pre-lethal changes in cell morphology may be enhanced. Also, these compounds are natural-like products and thus mimic the contents of the test library. There were no clear alterations in nuclear or cytosolic roundness between the treatments, and as can be seen from Figure A and B, changes in nuclear or cytosolic area were sufficient to identify abnormal cells. Indeed, doxorubicin increases both nuclear and cytosolic area, whilst Taxol did not significantly alter these parameters. Instead, it reduced cell number and increased the nuclear intensity, hence the third cutoff of mean DAPI intensity. Figure C shows the correlation between the nuclear and cytosolic area. The shaded areas indicate cells deemed to be abnormal. So abnormal cells have been defined as those of a cytosolic area of greater than or equal to 1,350 micrometers squared, or a nuclear area of less than 150 micrometers squared, or a mean DAPI intensity of over 1,500 units. These values may vary between cell lines, but can be easily changed in the assay. Thus, normal cells could be distinguished in Columbus, as highlighted in green, from abnormal cells in red, using the select population algorithm with the cutoffs manually inputted. A pair of abnormal cells have been circled in yellow to show detection by reduced nuclear area, object 2 in the table, or by increased nuclear DAPI intensity, object 3 in the table. The outputs generated from this analysis protocol are cell number per well and the percentage of abnormal cells per well, as shown by the heat maps on the right of this slide. The positive and negative controls, highlighted by yellow and red box respectively, can be easily distinguished from one another in terms of both cell number and percentage abnormal cells, positive controls being doxorubicin and 2 concentrations of Taxol. The 2 intraplate replicates are delineated by the blue line. Looking in more detail at the comparison between positive and negative controls, it can be seen that Taxol at a 2 micromolar concentration is very clearly distinct from the negative control of 0.2% DMSO. Whilst doxorubicin is less distinct, it is still statistically significantly different. The images in Figure C show the input image on the top line and those cells identified as abnormal highlighted in red on the bottom line. And you can see that there are more of them in the doxorubicin treatment and the Taxol treatment respectively in terms of cell percentage and number of cells. And also, mitotic cells were identified as abnormal in this assay, so it can also indicate changes in cell division as measured by a reduction in the percentage of abnormal cells. Following on from the development of this assay, it was important to assess if any of the cells identified as abnormal were apoptotic or necrotic. This was achieved using a Nexin-5 and propydium iodide staining. And indeed, a number of cells that would have been identified as abnormal in the Taxol-treated well are either a Nexin-5 positive, indicating they're in apoptosis, or a Nexin-5 and propydium iodide positive, indicating they're either necrotic or more likely dead, as highlighted in red in the right twomost images. Not all the abnormal cells would show up as apoptotic or necrotic, as shown by the cells ringed in the yellow circle, indicating the analysis package developed can identify subtle and prelethal changes to cell morphology, a valuable asset compared to cytotoxicity assessments based purely on cell counts. The utility of the assay was then assessed by Z prime factor calculations using the formula indicated. Z prime factors of between 0.5 and 1 indicate an excellent assay, whilst those between 0 and 0.5 a more marginal assay. The results of our Z prime calculations are shown in the table, with acceptable values of between 0.3 and 0.6 seen. As the test library has not previously been screened, in order to get an idea for false discovery rates, a number of test plates where the positive controls have been randomly arrayed were screened to calculate false positive and false negative rates. No false negatives were seen across these test plates, and our low false positive rates of less than 5% indicate we should have a low false discovery rate. Now that we have some degree of confidence in the robustness and specificity of the assay, the test library was screened in duplicate for each condition, with intraplate replicates generating 4 data points for each compound under the different conditions of 24 or 48 hours exposure. We identified hit compounds as those 2 or more standard deviations away from the mean of the vehicle controls, i.e. those compounds with a Z-score of greater than plus or minus 2. This cutoff was chosen as it correlates to a p-value of 0.045, and thus hits will be statistically significantly different from the negative controls. From the test library, 34 compounds were identified that reduced cell number after 48 hours by having a Z-score of less than -2. Those triangles below the lower dotted line shown on Figure A. No compounds increased cell number, and only one compound, number 137, also reduced cell number at 24 hours, as highlighted by the blue circles. This compound was also one of the 15 compounds to increase the percentage of abnormal cells at either time point. Those data points above the upper dotted line in Figure B. The assay also identified compounds which increased the percentage of abnormal cells without affecting cell number, e.g. compound 110, shown in the red circles. As follow-up to the hit identification, dose-response curves were generated for hit compounds, and examples for compound 137 are shown here. Figure A shows the changes in cell number that primarily occur after 48 hours exposure, except at the top dose of 20 micromolar at 24 hours, whilst Figure B shows alterations to the percentage of abnormal cells that increase with dose for both time points. Importantly, these changes, especially at 10 micromolar after 24 hours, show at least for this compound the assay has detected a pre-lethal change. Figure C shows example images of the abnormal cells identified for compounds 137 and 110. In the case of compound 110, it appears that the increase in abnormal cells may be due to alterations in mitosis, as a number of small cells are seen in close proximity to one another, frequently in pairs. So, in summary, I have presented here a very simple adjunct assay based on cell morphology for use with high-content screens to identify cytotoxic and pre-lethal effects during initial compound screening. This assay has the potential to be used in conjunction with hit selection from primary screens to reduce the number of compounds going forward for development that may subsequently fail due to toxicity. With further work, it may be possible to assign distinctive morphological changes to mechanisms of action. This has been discussed in the studies by O'Brien and Pearson that were looking at hepatotoxins by high-content screening. Indeed, this is another area where high-content screening is coming to the fore to streamline in vitro toxicity testing. Our work adds another string to the bow for the use of high-content imaging to detect cytotoxicity and reduce downstream compound failure. Thank you for listening. I would just like to thank the Bioscreening Technology Group and the University of Leeds for providing the facilities for doing this work, and the group of Professor Adam Nelson at the School of Chemistry for generating the test library, and the EPSRC for funding this work. Thank you.
And Heather, we want to thank you. Before we move on to Dr. Astori's presentation, I want to remind you once again about submitting questions through the Ask a Question box. Dr. Astori, the floor is yours.
In a recent guideline on the production of human cell-based medicinal products, the use of fetal bovine serum has been greatly discouraged by competent authorities. This because the risk of prions and other zoonoses, xenogenic immune reactions, variability due to seasonal regional differences, endotoxins and batch-to-batch variation, and we should also consider animal welfare concerns. It is possible to substitute the FBS with platelet lysate. This because platelet contains bioactive molecules and growth factors released by platelet organelles. In particular, PDGF and TGF are essential stimuli, and BFGF are essential stimuli for MSC's proliferation. Their inhibition reduced the MSC's growth by about 75%. All these molecules may influence mammalian cell proliferation and function, increasing the proliferation rate in comparison with FBS. So, human platelet lysate can be used to expand mesenchymal stromal cells. Mesenchymal stromal cells are stem cells first isolated from bone marrow by Friedenstein in 1974, are fibroblastoid cells, spindle-shaped, and they can differentiate into multiple lineage, and since they are stem cells, they are able to self-renew. MSCs have some important characteristics defined by the International Society of Cellular Therapy. Among them, they have to adhere to plastic. They express some surface markers like CD105, CD19, and CD73. They do not express hematopoietic markers like CD45 and CD34, CD14. And they are able to differentiate to the osteogenic, chondrogenic, and adipogenic lineage. Very important, they have immunomodulant properties in vivo and in vitro, and again, they are able to self-renew. The MSC sources, among them, the most important is the bone marrow, but MSCs are present also in adipose tissue, cord blood, and umbilical cord. In 2012, about 252 clinical trials were running all over the world. This to underline the importance of these cells in clinic. The platelet lysate is produced starting from blood donation or platelet apheresis. After a centrifugation step, the so-called platelet-rich plasma obtained after centrifugation is frozen, and the bags are then thawed to 37 degrees. And then, after another centrifugation step, platelet lysate is obtained. Our group have developed a fast production method for human platelet lysate by using ultrasounds. The platelet lysate is ready in 30 minutes instead of 24 or more hours. To do that, the platelet bags have been subjected to a cycle of ultrasound. The phenomenon of cavitation occurs when ultrasound hits the gas bubble in the fluid, and the compression and the rarefaction of the fluid makes bubbles contract and expand. As a consequence, if cells are present in the fluid, cavitation breaks cells membranes, and all the cytokines goes out of the cells, goes out of the platelets. In practical, PRP, platelet-rich plasma, was obtained from healthy donors. About 20 donations are pooled together and are subjected to ultrasonic treatment for 30 minutes at 20 kilohertz, and then the bags are centrifuged, the product is filtered and then stored to minus 80 degrees. We monitor platelet lysis by PDGF release. As you can see in graph B, the release of PDGF is proportional to the sonication time. After 30 minutes of sonication, about 74% of the PDGF contained in the platelets has been released in the media. And you can see from the FACS analysis in the upper part of the slide as the platelets are lysed along the process. We also had to test the efficiency of the platelet produced by sonication. To do that, we cultivate bone marrow mesenchymal stem cells in platelet lysate, and we induce those cells to differentiate throughout the osteogenic and adipogenic lineage. Not only, we were able to induce chondrogenic differentiation on those cells, and as you can see in the picture on the left part, you can see this little ball that is cartilage growth starting from the MSCs. We also tested the efficiency of the platelet lysate produced by sonication by measuring cell proliferation. Very interesting, you can see that expanding the cells in platelet lysate, in 10% platelet lysate, the mean cumulative population doubling time is about twice when compared to the cells growth in FBS. The immunophenotype of the MSCs is a typical immunophenotype of MSC cells, and also those cells were able to immunomodulate To do that, we expanded those cells in presence of platelet lysate. We blocked the MSCs, and the allogeneic lymphocytes were co-cultured in contact and transferred for 6 days. The cell proliferation was measured by bromodeoxyridine proliferation assays. As you can see to the graph, when we add the bone MSCs to T cells in a ratio of 1:5, the proliferation of T cells is almost completely inhibited. Safety testing is a very important issue, in particular for competent authorities. To check the chromosomal stability and genotoxicity of the cells growth in platelet lysate, we perform karyotyping at passage 7, and we perform micronucleus assay. This because we ask to ourselves if chromosomal instabilities may arise during a long-term culture, or if the increased proliferation rate could alter chromosomal stability of MSCs. For the micronucleus assay, we follow the OECD guideline. Micronuclei are chromosome fragments that lag behind anaphase during nuclear division. Micronuclei are biomarkers of chromosomal damage, genome instability, and cancer risk. The assay detects the clastogenic and aneugenic effect of substance in cells that have undergone division during or after the treatment with the substance under examination. The micronuclear assay is performed on the CHOK1 ovarian hamster cells. Those cells are very particular since this rodent cells lines is used for assessing genotoxicity potential in vivo. It is characterized by a mutation in the p53 protein that affects its functionality. It shows high sensitivity and low specificity for carcinogenity. In practical, we CHOK1 plate hamster cells in a black 96 well plate. Then at day 2, we add mitomycin or platelet lysate. At day 3, we block cytokinesis with cytochalasin. At day 4, we fix the cells with formaldehyde, and we stain the cells with Hux. And we perform image analysis. For the image analysis, we use a high-content screening technique. Usually, micronuclei scoring is performed manually on microscope slides, resulting in a between observer variability. The analysis is limited to 1,000 to 2,000 cells per replicate. And as micronuclei are relatively rare events, an accurate determination of their frequency requires examination of a higher number of cells. Usually, cells appear in many different morphologies after genotoxic or cytotoxic treatments, and this makes the analysis even more challenging. One approach to improve the efficiency of the process is the use of an automated image analysis system. For the latter, image analysis was performed by using an operator imaging system, Perkin-Elmer, and the analysis was performed using the micronuclei application in the Harmony software. There are 2 key steps. The first key step is the morphological identification of regularly shaped binucleated cells and the exclusion of irregular and isolated nuclei and detection of valid micronuclei. In this panel you can see in picture A that CHOK1 cells DNA was stained with UXT. In picture B, the cell nuclei were identified. In picture C, cell boundaries were marked. Picture D, micronuclei were selected as yellow dots within the cytoplasmic area. In image A, only valid micronuclei were detected on the basis of location, dimension, and intensity. And at the end, in image F, only micronuclei within the cytoplasm of binucleated cells were scored. Coming to the data analysis, cytotoxicity was estimated by the evaluation of the reduction of cell number after treatment and by the cytokinesis block proliferation index. The quality of the assay was evaluated by calculating the signal to background ratio and the zeta factor. The result obtained at the acquisition condition of 10x and 20x were compared. I have to mention that all these analyses were performed at University Trento at the Center for Integrative Biology. As a result, you can see in the graph A the histogram representing a dose-response curve for the known genotoxic agent mitomycin A. You can see that there is you can see the proportionality between the number, the percentage of micronuclei formation, and the concentration of mitomycin. In the graph B, you can see the histogram representing the effect of platelet lysate on the hamster cell line, and you can see that none of the tested concentration of platelet lysate was able to produce micronucleation in the cells. Even at very high platelet lysate concentration, about 30%, the percentage of micronucleation didn't change significantly compared to controls. These are the results of the image scoring with 10x and 20x objective. The most robust assay was obtained using the 20x magnification. Slightly lower values were associated with the 10x magnification, but the larger field of analysis allowed for a reduction of the acquisition time of about 33% of time. The safety of platelet lysate was monitored by karyotyping the cells at P7, and the karyotype didn't reveal genomic alteration on bone marrow's MSCs expanded in platelet lysate. Coming to the conclusion, we have automated the micronuclei scoring, allowing for a fast and reliable acquisition of data compared to visual inspection of slides. In conjunction with HES technique, the results were improved with respect to robustness, power, speed, and accuracy. The 20x objective gave the best results because of a high resolution, and the acquisition by using the 10x magnification resulted in a reduced micronuclei discrimination ability, but had the greater advantage of reducing the time for the acquisition from about 50 to 70 minutes for a complete 96-well plate. Platelet lysate is a valid substitute for FBS. This because can be produced efficiently by sonication. The cell proliferation is significantly faster compared to cells expanded in FBS. The platelet lysate doesn't induce genome instability on MSCs at P7 by karyotyping cells, and platelet lysate doesn't induce micronuclei formation on HOK1 cell line. And thank you for your attention.
Thank you, Dr. Astori. Before we start the Q&A session, I also want to give a final call on the questions for our panelists. Doctors Martin and Astori will be joined by Dr. Valentino Adami from Sibio, Uni Trenti. He's also very familiar with HCS analysis and will help answer your questions. So feel free to submit all of your questions since we have 3 experts available to answer them. I also want to let you know that a post-webinar survey will deploy shortly in the presentation manager. You might need to disable your pop-up blockers in order to view it, but please do take a few minutes and respond. Your comments are extremely valuable to us. Okay, we're ready to start our Q&A discussion. We have a lot of good questions already, and we'll answer as many as we can. Our first question is for Heather. Heather, as someone new in the field, it would help me if you could talk to the novel aspects of your assay. How is this different to other assays typically used?
Okay, so the first key point about our assays, although we're looking at toxicity, we're kind of designed the assay so it could be used really in conjunction with activity assays, so it doesn't, you don't have to do 2 separate assays. It's novel because it doesn't just measure cell number. You also get changes in morphology using a far-red marker that's not typically used for this. So it's a little bit different in that respect, and although it's designed to measure toxicity, it's designed not to impinge on other activity-based measurements by high-content imaging.
All right, Heather. What percent DSMO did you use in the first experiment, and how did DMSO compare to a no DMSO control?
Okay, all experiments were done with 0.2% DMSO, so it was constant across the plates. And initially we did some experiments, and this level of DMSO has absolutely no effect compared to a no DMSO control. The numbers for all the outputs were identical. So.
All right, Heather, here's another one for you. How many plates, 96 well or 384 well plates, can you screen per day? How many minutes do you need to measure one 96 well plate or a 384 well plate?
Okay, so our assay actually takes place over sort of 4 days because it's got an incubation time, but we sort of do this in usually between 10 and 20 plate batches at a time, just because that's a nice number to handle for our robots and stuff. And the amount of time needed to measure one 96 well plate or one 384 well plate in our assay, it depends on the magnification we're using and the number of fields we're imaging. In the particular assay I talked about, it takes about 50 minutes to an hour to image a plate, but that's 3 channels and 12 fields of view per well, so we're getting quite a lot of data from that.
All right, Heather, thank you. Giuseppe, I have a few questions for you. The first one, did you try to induce micronucleation by using MSC or other cell types?
Yes, sure. We try to induce in parallel with hamster cells. We try to induce micronucleation both on MSC cells and on fibroblasts. And it was very interesting because when we add the mitomycin, even at very high concentration, those cells were not able to show any formation of micronuclei. And this is in particular very interesting for MSCs because suggests an important repair mechanism of those cells, so those stem cells. And this for sure would represent a more physiologic model to test the effect of platelet lysate. But at the end of the story, we were unable to induce any micronucleation. So the signal window between the control and the induced cells was too low to make any final conclusion.
Okay, thank you. Here's another question. How many cells did you image, sample, and why?
Okay, I will answer to this. We imaged and analyzed at least 2,000 binucleated cells per concentration, according to the OECD guidelines for testing chemicals.
Okay, thank you. Here's another one. Have you been using high content analysis to follow MSC differentiation?
Very, very, very important, interesting question. Yes, actually, we follow the stem cell differentiation by normal microscopy, in particular for the differentiation of for the adipogenic differentiation. What we do now is just to count the number of the droplets of fat that are forming in the cell. But the quantification of the differentiation is a very important issue, since we know that different clones, different MSCs coming from different tissues have a very different differentiation ability. It is important for example, if we want for tissue generation issues. And I know that with the operetta system it's possible to follow that, to quantify the differentiation. Also this device allows us to, for example, investigate if there are some modification in the cytoskeleton. So this is, I think that a first next step in our research would be to try to quantify the differentiation of the cells by using the operetta.
Thank you, Giuseppe. Heather, I have a few more questions for you. Do you think the same parameters could be used with other screening cell lines to identify cytotoxic effects?
I certainly think the same measurements could be used. Whether the actual numbers were the same is probably unlikely because every cell line is slightly different and you might need to change the size cutoffs. But in principle, I think it could work. We have used it with a few other cell lines that haven't made it into publication or anything yet, so I think it's distinctly possible.
All right, Heather, here's another one. Have you tried other parameters than those described, for example, texture or star morphology to identify toxic compounds?
Okay, so these are assay aspects within the operator analysis protocol programs. We had a look at them. We decided they gave it, in terms of what we wanted to get out of this assay, we wanted it quite simplistic. So we decided these outputs were actually quite complex in terms of what we were getting out and interpreting them. So we decided we would keep it simple because it is basically an add-in to activity assays rather than a standalone toxicity assay.
Thanks, Heather. Giuseppe, I have another question for you. First, a compliment. They said, great data and compelling evidence that the HCS approach is an improvement. Can you talk to the value of the MN assay compared to other genotox assays such as GADD45A?
I have to admit that the question is very interesting, but I don't know this GADD45A assay. I cannot answer to this question, sorry.
Oh, I'm sorry about that. Unfortunately, our time is up. Thanks for listening in, and thanks for the great questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists and our guest question answerer, Dr. Valentina. Thank you for joining us, as well as our sponsor, PerkinElmer. Thank you for your time today.
Thank you.
Thank you.
Thank you.